There are a variety of changes that occur in tissue after death. Pathologists want to study these tissues in a form that is as close as possible to what they looked like in their living state. Thus, as quickly as possible, tissue samples must be fixed, which means that the processes that naturally occur in decaying tissues are halted. Once tissue has been fixed, it can be grossed or cut up into representative samples and placed in plastic trays called cassettes. These cassettes run through special machinery that hardens them either by flash freezing them or infusing them with wax before a pathologist would cut them into thin slices only 1 cell thick for microscopic analysis. Here is where that process begins. Here I am setting up my working station, including the tools I will be using during the dissection, formalin, a dissection tray, and the specimen, which is a kidney in this particular instance. I start by setting a container of formalin, which is a fixative solution, under a fume hood since we are dealing with some pretty strong chemicals. I place the lid on the formalin container and label it with patient information, including the name and the date of birth of the patient. I then submerge the organ samples into the fixative solution and swirl around the container to ensure the tissue sample does not stick to it or any of the other specimens. I replace the cap over the container after each tissue is added to the formalin, as one container can be used for several different types of tissue samples. For example, you can see I also add an eyeball specimen to the jar. After that, I write a label on the container naming the tissue types I have added to it. After the fixation period, which varies from tissue to tissue, I remove the sample from the formalin container and place the kidney on the dissection tray, once again replacing the lid back on the fixative container to avoid unnecessary exposure to the chemicals. I start grossing the kidney by slicing it in half to facilitate further dissection and make the sections smaller. I cut a few pieces of tissue from different locations on the organ to ensure that I will have a variety of cell types to analyze. I then place the pieces into plastic cassettes and put the lid onto the cassette. Next, I label each cassette with the patient's name and date of birth and permanent marker to avoid smudging the ink after processing. Lastly, I store the cassettes in a formalin container until the cassettes are ready to be put into the paraffin wax machine.